Welcome student in the second lecture of our discussion about anomalous structure. We have discussed the presence of accessory structures that were formed before secondary growth formation. Now we will discuss those structures which arise after secondary growth formation, and there are different conditions by which these structures are formed. Now one way is the presence of normal cambium with abnormal activity. That means the cambium is normal, that it is present between vascular tissue. Okay, and it's also important to note here that mostly we are talking about those structures which are giving rise from the vascular cambium. Okay, because that is the normal behavior. However, when we talk about core cambium, that can arise from different locations. Okay, so we are generally talking about the behavior of a normal vascular cambium. Now, in this cambium is present. Okay, generally it is found in dicot plants in between the phloem and xylem. So this you all know now. So this cambium, what it will cause? So there are multiple vascular bundles. So this cambium will form a ring-like structure, interfascicular and intrafascicular. Both the cambiums will join together and they will form a ring-like structure, giving rise to xylem on the lower side and a phloem, continuous ring of phloem on the outer side. See, normally this is the pattern, and this also that the amount of xylem is cut more as compared to secondary phloem. So in this case also there can be different variations. In one, the cambium which is forming vascular tissue only in the region of vascular bundle. See, normally they are doing so, but sometimes some cases can be there that this cambium, okay, they are giving rise to vascular tissue only in the fascicular region, okay, in the vascular bundle region. So this case is common in Cucurbita, vitis and Aristolochia, okay, these species. Whereas in some other, cambium is forming a ring-like structure, but it is giving rise to xylem and phloem in different proportions, okay. That means the amount of xylem which should be actually formed is not as such, or the amount of phloem which should be formed should not be as such, and they are also oriented in different ways. And sometimes they also give rise to parenchyma in xylem. So this is also one of the anomalous behavior that we will study. So first of all, let's try to understand this behavior, that it is forming vascular tissue only in the region of vascular bundle. See, in this structure you will see in Cucurbita. See, I have told you before that many of the structures which are found, or many of the plants have multiple anomalous behavior. Okay, it's not the presence of only one thing is sufficient to describe that anomalous structure. So they will have multiple. We will try to understand them. See, in Cucurbita, we have seen that in the previous class that it was having two rings of vascular bundles. Okay. So in this case they were having wavy margins, and at the ridges there was collenchyma, and in the center you will have that pith and pericycle cortex, that all you will have it. In the center some big vascular bundles, inner layer of bigger vascular bundle and outer layer of smaller vascular bundle. And they were collateral. See, this is also very important to remember. Because this is also not a normal feature. So what happens? In this, out of the two cambium, only the outer one becomes active. Okay, although there are two cambium, but the inner one is not active and it is not taking part in secondary growth, whereas the outer one is active. And what it does, now the outer cambium will join together the interfascicular region between this cambia will become active, and they will also form a wavy ring-like structure, because see, now since they are orientation in different different locations, they are not on the same plane, on the same diameter. So a wave, first of all, you will see that a wavy outline of this cambia is formed. Cambium ring, you can say, but this cambium ring is not acting in normal way. Normally it should produce xylem on all the sides and phloem on the outer side, but what it is doing, only in the fascicular region. Okay, this cambia is cutting xylem on the inner side and phloem on the outer side. Okay, suppose this is the, so this cambia is forming secondary xylem on the lower side and secondary phloem on the outer side. So what is observed, that this vascular bundle just increases in size. Okay, it's not that you will not find any continuous ring, only some small increase in vascular bundle is seen, and in this you can see here the presence of secondary xylem, and primary xylem is towards the lower end. Okay, this is the primary xylem towards the lower end. Okay, and secondary xylem, and even here also you will find secondary phloem, and say primary phloem might be crushed because of the increase in somewhat diameter of the vascular bundle. Now next is Aristolochia stem. In this also what you will see in this normal vascular bundles will be there. See, in this case there isn't any anomaly which is found before secondary growth formation. That means before secondary growth formation there will be a normal ring of vascular tissue. And you will have endodermis, pericycle, all those things will be there. So you will see normal vascular bundles arranged in the form of ring. Okay. So now when cambium will be formed, cambium ring will be formed, it will join in normal fashion. As we have seen in cucurbita, that was having a wavy margin because of anomaly in the orientation of the vascular bundles, but in this case you will find a normal cambium ring. But in this also, this normal cambium will still behave abnormally. What it will do, it will form vascular bundles only in the region of a fascicular region. Okay, it will give rise to secondary tissues only in the region of fascicular vascular region. Okay, suppose this is the cambium and it is increasing xylem here and phloem on the outer side, and on this side they will produce parenchyma on both the sides. So what will happen? There will be an increase in the vascular bundle. Okay, you will have secondary phloem and a small amount of primary phloem. And this is the secondary phloem. And here also you will see most of the things will be secondary xylem. Okay, and small amount of primary xylem will be left in the end. So in this case also you will still find discrete patches of vascular bundles, as you have seen in cucurbita also, that there is no continuous ring formation because the center region is occupied by parenchyma. So as discrete patches of vascular bundle were there in the primary structure, similarly you will also see those discrete patches in the secondary structure also. See, this is the diagram of Aristolochia stem, okay, cellular diagram, where you can see cortex, then endodermis, pericycle, then phloem. Okay, this is basically secondary phloem will be there and primary phloem will be small amount will be crushed on the top. Then you have cambium, then secondary xylem will be there, and then primary xylem. In between parenchymatous medullary rays. Okay, that will give rise to medullary rays that we have discussed before, that medullary rays are found between vascular tissue. Okay, because since there are big patches of parenchyma, so these bundles will appear discrete. Otherwise, in continuous patches also single small rays of medullary rays are observed. Here see, you can see here some periderm formation. So that we have also discussed that when a stem is thicker and they have wider diameters, okay, when they are hard, they form periderm. So in this case also a periderm formation is there, cork layers are there, cork cambium is there, and lenticels are also coming out. So depending on the age of the plant, depending on the structure and size of the plant, the periderm layer will also be formed. See, you can see very thin periderm or you can see a very thick periderm also. Okay. Now coming to the next unusual behavior of cambium during secondary growth formation, which is formation of unusual proportion of xylem and phloem, in which a continuous cambium ring is formed. They are continuously giving rise to xylem and phloem, but different proportions of xylem and phloem is formed. So the first important example is bignonia. Bignonia stem. See, bignonia, they are also, like you can say, they are creepers and or lianas, you can say climbing plants. So many a times you will see such type of anomalous behavior in lianas, because that might help them in having some support or having some advantage during their growth. So in bignonia also, it is also known as cross vine. Okay. Now in bignonia, what happens? You will have a normal, initially everything will start with a normal structure, okay. Normal ring of vascular bundle, okay. This vascular bundle, in this cambium will join all the intrafascicular cambium and interfascicular cambium, they will join together, okay, and they will form a ring like structure. They will form secondary xylem and secondary phloem, okay. Suppose this is the fifth region, they are cutting secondary xylem, normally they are cutting, okay, and phloem also they are forming. This is the secondary phloem. But after some times of this normal behavior, this cambia starts behaving abnormally. What it does at certain points, at certain regions, see, cambia is a single layer structure, okay, although it might appear multiple layer, that also we have discussed because of formation of some cambium initials, but generally it is a single strip of meristematic cells. So at certain strips, okay, or at certain locations, it will stop its activity, okay. Rest of the cambia will start functioning normally, but at certain points, see, actually I cannot say that it will stop activity, it will behave abnormally and it will cut xylem less as compared to phloem. So it will cut more phloem as compared to xylem, okay. So it's not inactive, it is cutting more phloem as compared to xylem. So in bignonia at four corners, okay, generally just diagonally opposite to each other at four corners, what will happen when this cambium will behave like this? So okay, so in this way we will see presence of phloem wedges, okay. Sorry. Okay, so these are phloem wedges. That means extension of some phloem inside the secondary xylem. So this is the secondary xylem, okay, and this is an entire part of secondary phloem. See, the outer one which is forming the ring is also secondary phloem, and this one which is entering in the form of wedge is also secondary phloem. It is just an extension because at this point more phloem was produced by cambia as compared to xylem. So that's why an intrusion or a phloem has intruded inside the xylem in the form of wedge shape. Now in the phloem wedge also you will see bast fibers. See, remember that in phloem you have fibers also, okay. So those fibers we generally call as bast fibers. So even when you will observe the transverse section and you will stain them, you will see some pink color structures in this phloem also, and these will be bast fibers. Okay, so you can see these 4 phloem wedges because in Bignonia in a cross shape you can say at diagonally opposite to each other 4 phloem wedges will be formed. Now later in much older plant more 4 more wedges could also be again they will start its formation, and now you can see even 8 wedges, okay. Sometimes 8 wedges could also be seen, but they will be also diagonally opposite to each other. So like in this, so in this way you can see this all 8 wedges. So these are phloem wedge, okay. Okay. And this is xylem. So this is all secondary xylem and this is all secondary phloem. So in Bignonia you will see this type of structure. So this is also a TS of Bignonia stem which I have prepared this slide to show you that this is just one wedge, otherwise on all other 4 corners 4 similar structures would have been formed. So in this you can easily say that this is the pith region, and above that you have secondary xylem, okay. Primary xylem is definitely below, but you cannot differentiate them, and this is all phloem. See, this is also secondary phloem, but this is an extension of this phloem inside xylem, and this is known as phloem wedge. Okay, you can see bast fibers also. Similarly, some sclerenchymatous patches, suppose in this case the pericycle was made up of sclerenchyma, and when it is getting crushed and when some sclerenchymatous patches also you can see here which is of pink color. See, this is not secondary tissue, okay. In this you can see some chlorenchyma also if the stem is performing photosynthesis. You can see some periderm also coming out. This is the normal structure, but all these structures are normal. This is also the cellular diagram which is showing the same structure. Now the cambia is active here, the cambia is active here, okay. Sometimes the wedges are exactly rectangular, sometimes they will be slightly wider on the upper side and lower on the shorter on the lower side, okay. Then Bauhinia stem. Bauhinia is also a clina, okay, it is also a climber. So in this case also you have a cambium which is behaving abnormally, okay. Suppose cambium ring is formed in this case, and in this case cambium is active only on 2 sides. So if it is active on 2 sides, definitely it will form secondary tissue in this direction and in this direction. So it will form like a plate or a band or strap like structure, and this will actually help them maybe in climbing. Okay. So the stem will be flat because it is just growing on two sides. You can see secondary xylem and a small patch of secondary phloem. Okay. So in some species of Bauhinia, this type of they become flat or strap like, whereas in some species, in other species of Bauhinia, it uh, forms ridged structure. See, in this case, cambium is forming tissues on all the side, but still it is inactive or it is giving rise to tissues in different proportion on at certain points. So it is forming ridge like structures. Okay. And this ridge like structure is definitely due to abnormal activity of the cambium. Now another case is there in which this xylem forms a island of parenchyma. Okay. Now in this case also it is found in Urtica dioica stem. So in this case also they will definitely have a normal ring of cambium. Okay. They will form they are forming tissues. Okay. They are cambium is forming a ring of cambium, and they are forming tissue. Suppose this is the cambium. Okay. It is giving xylem. Okay. It is forming xylem, but at certain points what will happen, this cambium will start giving rise to phloem. On the outer side it is normal, but it will start giving rise to phloem. Sorry, it will start forming parenchyma. This cambium will start forming parenchyma. Okay. And again after forming parenchyma for some time, it will again form xylem. So in this way a parenchymatous patch will be embedded inside the xylem. So when we will observe the transverse section of this plant, we can see lot of parenchymatous tissues embedded in it. And how you can make out that this is parenchymatous tissue? See, if you will stain them, definitely all the tissues of the xylem will be stained, but this parenchyma will not be stained. If suppose if I want to stain them with fluoroglucinol, which will stain the tissues which have lignin, so parenchyma will not be stained. So in that case you will see lot of parenchyma unlignified parenchyma will be embedded in the xylem. So they will appear as islands of xylem which are present in a lot of patches of parenchyma which is present in xylem. Okay, so this is also another abnormal behavior of cambium. So with this I end up here, and in next section we will, or in next lecture we will try to understand many more abnormalities which arises during secondary growth. So thank you for today.